Hi, I'm Gleb Goussarov. I'm affiliated with the Belgian Nuclear Research Center and Ghent University, also from Belgium. Now I will present our project to use oligonucleotide frequencies to evaluate the quality of metagenomic bins. Let's begin with a bit of context. What you see on this slide is a predictive capability of the most commonly used tool to predict metagenome bin quality, a tool called CheckM, which relies on the presence or absence of universal marker genes. You can see that it is quite decent at predicting completeness and not that great at predicting purity, at least using our own datasets. So we wanted to try and see if we could use another approach. The approach we have decided upon is to use oligonucleotide frequency signatures, which I will now briefly explain. Here you have a nucleotide sequence, which we split into shorter oligonucleotides of a given length, say 2. We count them and then examine the relative abundance of each oligonucleotide. The resulting vector is what we refer to as a genomic signature. Previously, we have used a similar approach to calculate a distance between genomes with a novel method we called PASSIT4. The distance was particularly efficient at identifying whether or not two genomes belong to the same species or not, as illustrated on the figure here on the right. We wanted to see if we couldn't use a similar approach to help with binning metagenomic contigs. The idea is sound in theory, since individual genomes tend to have relatively stable signature depending on the window choice. If we use a very short window, like the one here with 1 kb, then there is still quite a lot of variation within signatures. But if we use a long window, then the signature is pretty much homogeneous throughout entire bacterial genomes. So we began by trying to find the best window length to use for different signatures. With a length of 1 kb, different species are hardly distinguishable from each other. But with a length of 50 kb, the species can be distinguished from each other relatively well. Unfortunately, using our own approach mentioned in the previous slide, we found that it doesn't quite work out, because even at 50 kb, the variance is still not enough to distinguish between species. So in order to maximize our chances of success when building a new model, we have included a number of different signatures using raw frequencies, but also a Markov model Z-score approach and our modified PASSIT approach. We computed all against all distances within a given bin to estimate its quality. As a result, for each bin, we obtain a distribution like the one you see here. To make it usable for a model, we compute a number of statistics to feed it. So these are things like average, standard deviation, and so on. Here's what we have produced in terms of results. We have two sets of models, one of which relies just on distribution statistics, and one which also takes into account the results of marker gene-based tools, in this case CheckM. The tool, which relies purely on distribution statistics, is still underperforming at the moment, and the tool that also relies on CheckM actually shows a marginal improvement over just using purely marker gene-based approach. As we are still in the process of improving our models, there is not yet currently available, since we do not explore the number of potential avenues for improvement. However, once finished, we will provide the preprocessing tools that we have developed specifically for preprocessing assemblies of metagenomes, the final models we have generated in both an R data format and as a standalone, and we will ensure that these can be used across multiple operating systems. Thank you for your attention. If you found this topic interesting or would like more information, you can always contact me at the address that has been listed at the top of each slide. The poster number, if you want to speak with me personally, is number 41. Thank you for your attention again.